So my presentation is over tight junctions. So some overview and general information. The tight junction is also known as the zonula occludens. It leaves little space between the plasma membranes of adjacent cells. The membranes of the two cells come very close together, and the outer leaflet of both membranes appear to be fused. Protein components are arranged like beads on a string. They act as a barrier, preventing material from passing between the interacting cells. They're very, very, very close together. The bands will form a barrier that will only let select ions and molecules pass between the cells. Most molecules are required to go through the cell, allowing the cell to regulate its passage through the use of transporter proteins. Structure and various proteins involved. Each junction has a series of ridges. So if you look at the first part of the picture, you can kind of see the ridges along the plasma membranes. The membranes are connected at points of contact along the ridges. So if you see the TJ protein complex, it's going to point out on the first side of the picture. You can see where they're actually connected along the ridges. Each ridge has a continuous row of packed transmembrane proteins, about 3 to 4 nanometers in diameter. There's more than 40 different proteins involved in the tight junctions, including transmembrane and cytoplasmic proteins. The TJs recruit cytoskeleton proteins such as F-actin. There are 3 major transmembrane proteins involved, including occludin, claudins, and junction adhesion molecules, which are also referred to as JAMs. So if you look at the second part of the figure, we can see where there's 2 cells, cell 1 and cell 2, and the membranes are becoming the junction is forming. So if you see at the top, the JAM transmembrane protein is going to be at the top in the orange, occludin is going to be the light blue, and then the claudins. And then we can also see a lot of other components that take place for this junction to occur. So the location in an organism or cell I chose were the tight junctions that are found in human intestinal epithelial cells. The tight junctions are the key structures that regulate paracellular movement of macromolecules. The permeability of the intestine actually allows fluid and solute exchange between the lumen and select tissues. So the tight junctions need to maintain this barrier in order to prevent the permeation of pathogens, toxins, and antigens into mucosal tissue and the circulatory system, because somebody could end up very sick if these junctions weren't working properly. Several factors are needed, however, for the tight junctions to help regulate, such as the absorption of water, nutrients, certain ions, everything that's needed. A disease I chose was inflammatory bowel disease. These include Crohn's disease and ulcerative colitis. It affects 1.4 million Americans. Most patients have increased intestinal paracellular permeability, which will ultimately slow the decrease of their epithelial barrier function. So they're going to have a lot of toxins coming into their mucosal areas. The decrease in the barrier function actually suggests that there was a loss of the tight junction. It causes the mucosal immune system activation to become altered, which is what they're stating is the major cause of Crohn's disease, ulcerative colitis, other inflammatory bowel diseases. I also read a study where they're saying that therapies now are being used to target the restoration of barrier function in order to help slow down the progression of this disease rather than alternate therapies they were using in the past. Some recent findings, I've seen that there have been several advancements in the understanding of tight junction functions, composition, and regulation. One that I thought was very interesting was that they say tight junctions are actually in human epidermis. It was debated for several years. The original concept they stated was that paracellular flux of several factors was actually only regulated by the intracellular lipids present. Electron microscopy was actually used to show that intracellular junctions morphologically were there, and they were morphologically compatible with tight junctions. Conclusive evidence of epidermal TJs was established actually at the beginning of the millennium. However, new findings are further supporting this. Tight junctions have been found to play a role in the cutaneous immune system through innate innate and adaptive immune barrier, which are both very important. So my question would be, what type of tight junctions, what type of cells that have tight junctions do you believe make up the blood-brain barrier? And if these tight junctions failed, what could potentially happen to somebody?